Ocular Revision is an artwork using an alternate mechanism for the analysis and display of the DNA image. This novel circular apparatus is designed as a focal point for reflecting upon a shift in the life sciences from the study of life itself to a more reductive focus upon non-living DNA. Biology, which defines the cell as the basic unit of life, is a discipline broadly impacted by an ocular frenzy of the modern period. Optical devices of this period include telescopes, microscopes, endoscopes, gastroscopes, oscilloscopes, and a multitude of related visual tools. The visual information produced by these devices was based around the lens, which, like the eye that it aided, was fundamentally circular. Conversely, the DNA image belongs to a different regime of late 20th century screen-based information: Cartesian coordinates, the grid, and the database. And while the 19th century marks the age of biology, the late 20th century marks the post-biological age, because DNA is increasingly understood as a code rather than a material substance. The post-biological turn marks a point of cybernetic fantasy in which the infinite complexity of the wet organism is finally humbled by the easily quantifiable genetic code that supposedly exercises total control over the flesh. Ocular Revision is an artwork using an alternate mechanism for the analysis and display of the DNA image. Typically, DNA is visualized in a rectangular chamber containing a porous gelatin that has an electrical field across it. When DNA is inserted into one side of this gelatin, the electrical current pulls it across the gel at a rate corresponding to its molecular mass, and thus differentiates DNA of different sizes. Ocular Revision uses a custom experimental circular gel electrophoresis rig to visualize DNA bands. This circular rig was designed to be polarized from inside to outside of the circle. The radial design and the inside-out polarization allows the apparatus to create DNA image reminiscent not of a bar chart or a progress bar on a computer screen, but rather a slow emergence, a signal, a flowering, an attraction or repulsion. The work also inverts the interpretation of a key trope used in contemporary genomics and the basis of the terms genetic map or DNA mapping. What the terms refer to is experimentally locating specific genes within DNA sequences, making maps of DNA. DNA is the subject. In Ocular Revision, on the other hand, I make maps with DNA, DNA as medium, not subject. By selectively processing DNA of common laboratory organisms, I seek to create DNA images resembling hemispheric images of the Earth's continents. The current video clip shows time-lapse footage of the creation of the Western Hemisphere, playfully challenging use of such a trope as mapping, because mapping implies distance simplification, abstraction, and exploitation. For example, political and economic maps of the world. Ocular Revision uses amplified DNA segments from the E. coli K12 strain, a safe molecular workhorse since the 1950s which cannot live outside the laboratory. First, the bacteria is incubated into colonies. Then DNA is extracted and purified. Then custom primers are designed by targeting the appropriate DNA regions in its genome. These primers can amplify correct-sized fragments from the organism to behave correctly in the gel. Ocular Revision is visualized upon twin circular projection screens flanking the instrument cabinet, which houses electronic components for electrically polarizing and blue light transilluminating the electrophoresis gel. The clear acrylic electrophoresis rig rests atop the instrument cabinet, and varied imaging equipment can be attached above it. According to Michel Foucault, the birth of biology, around 1800, corresponds to a new discursive object, life itself. Conversely, the object of post-biology is non-living DNA, abstracted as pure code. Furthermore, the post-biological age is not an age characterized by the eye and perception, but by the database and informatics. suspicious of many of the convenient and pervasive computational metaphors operationalized in post-biological genomics. With ocular revision, I reassert the physicality of DNA as a substance and medium, neither a command code nor a call to destiny, but a substance deeply enmeshed in the matrix of the living, that is not virtual nor merely symbolic, but continuous with the deep materiality of life and living processes.